Today's lecture will be on introduction, which is the most rapid and oldest method of plant breeding. If we go into the details of this method of breeding, plant introduction according to Allard in the year 1960 is the oldest and the most rapid method of crop improvement. Introduction basically it's a process of introducing or domesticating crop plants into new area. Introduction may involve domestication of new varieties of the crop already grown in the area or wild relatives of the crop species or even totally new crop species. Example, contender variety of French bean. And plant introduction can be within a country, between the states, or between the countries, or even between continents. Introduction is basically classified into two types. One is primary introduction, the other one is secondary introduction. Primary introduction is nothing but when the introduced variety is well suited to the new environment and is directly released for commercial cultivation without any change in the original genotype is known as primary introduction. Example is IR8 variety of rice and Sonara 64, which is a semi dwarf wheat variety. And if we come to the secondary introduction is nothing but when the introduced variety is subjected to selection in order to isolate superior variety out of it, or may be hybridized with local variety in order to transfer one or few desirable characters to the local variety is known as secondary introduction. Example is Kalyan Sona and Sonalika varieties which are selected from the material introduced from Summit, Mexico. And next, there are various agencies which are involved in plant introduction. And however, among all the agencies noted so far, NBPGR works very much prominently in this area of introduction, plant introduction. And this NBPGR, National Bureau of Plant Genetic Resources, which was organized in 1976, is mainly responsible for introduction and maintenance of germplasm of agriculture and horticultural crops. In addition to National Bureau of Plant Genetic Resources, there are also some other agencies which are concerned with plant introduction, like the Forest Research Institute of Dehradun, which is responsible for introduction, maintenance, and testing of germplasm of forest trees. And next, the Botanical Survey of India, which was established in the year 1890, is responsible for introduction, testing, and maintenance of plant materials of botanical and also medicinal interest. But at present, introduction and improvement of medicinal plants is being done by NBPGR itself. And next, the Central Research Institute for various crops like tea, coffee, sugarcane, tobacco, potato, rice, etc. This institute, it introduces, tests, and maintains plant materials of their interest. But their activities are regularly and timely on a timely basis. They are coordinated by NBPGR, which has the ultimate responsibility for these introduction activities. Plant material may also be introduced by individual scientists, universities, and also other research organizations. But all the introductions in India must be routed through NBPGR, New Delhi. And if we go into the details of these introduction program or crop improvement program of introduction, the main stage is given to NBPGR. And this NBPGR, it has its headquarters located in New Delhi, IIRI, India. And it has four substations basically for testing of introduced plant materials. These substations, they represent various climatic zones of India. and they are listed as first one Shimla, and it is situated in Himachal Pradesh and it represents the temperate zone. And next, it is approximately about 2300 meters above mean sea level. Next, Jodhpur of Rajasthan, which represents the arid zone, and Kanyakumari of Tamil Nadu represents the tropical zone, and Akola of Maharashtra represents mixed climatic zone. And this Akola of Maharashtra, which represents the mixed climatic zone, it was recently shifted from Amravati. Previously, it was located in Amravati, and now it is shifted from Amravati to Akola. And there's a special um, zone which was recently established. It's a new substation in addition to the above four substations. It's located at Shillong and it's mainly for the collection of germplasm from northeastern India. And this part of the country has a large genetic variability for several crop species like rice, citrus, maize, etc., mainly because of the varied climatic condition in that area. And now, if we look into the activities of this NBPGR, National Bureau of Plant Genetic Resources, it has been doing a great effort in conserving, and also supplying this plant genetic material. And it basically functions as a central agency for the export and introduction of germplasm, which is mainly of economic importance. And this bureau is assisted in its activities by various central research institutes of ICAR. And the activities of this bureau can be summarized like first, introduction. It introduces the required germplasm from its counterparts or other agencies in other countries, and also it arranges exploration inside and outside the country in order to collect valuable germplasm, and it is responsible for the inspection and quarantine of all the introduced plant materials. The next, testing, multiplication and maintenance of germplasm obtained through various sources. This may be done by the bureau itself at one of its substations or by one of the concerned central institutes of ICR also. And next, to supply on request germplasm to various scientists or institutions. And the germplasm may be supplied ex stock or may be produced from outside in case it is not available in the country. And next, maintenance of records of plant name, variety name, propagating material, special character, source, date, and other relevant information about the materials received. And next, the main one is to supply germplasm to its counterparts. Introduction from the counterparts, and again supply of the germplasm to the counterparts or agencies in some other countries. And next, to publish its exchange and collection lists. Whatever it has exchanged, whatever it has collected, it maintains lists and records, and it regularly publishes this exchange and collection lists. An introduction newsletter containing such lists is being published by FAO, Food and Agriculture Organization, since 1957. It's being done in regular intervals. And NBPCR also published some lists and is in the process of publishing some other catalogs also. And next one is to set up natural gene sanctuaries of plants where genetic resources are endangered. And also it has another important activity, that is improvement of certain plants like medicinal and aromatic plants. Medicinal and aromatic plant improvement is also being done by this agency. And if we go into the details of the procedure of introduction mainly has six steps, important steps. And the first one is obviously procurement of the genetic material, that is germplasm. Any individual or institution can introduce germplasm in India, but all introductions must be routed through NBPGR, New Delhi. And the next one is quarantine. Quarantine means in order to keep the materials in isolation in order to prevent spread of diseases and all. And all the introduced plant propagules are thoroughly inspected for contamination with weeds, diseases, and also insect pests. And next one is cataloging. This cataloging, basically when an introduction is received, it is given an entry number and further information regarding name of the species, variety, etc., etc., and these are recorded. and in cataloging these plant genetic material again classified for catalog, so that for recording purpose and also for reviewing it, in order to make that work easy, they're again classified as exotic collections, indigenous collections, and indigenous wild collections. Exotic collections are given the prefix EC, capital E, capital C. Indigenous collections are designated as IC, capital I, capital C. And indigenous wild collections are marked as IW, capital I, capital W. And the next step in the introduction procedure is evaluation. Evaluation: in order to assess the potential of new introductions, their performance is evaluated at different substations of the Bureau. And the next one is acclimatization. Acclimatization is nothing but its adaptation to the new environment. And generally, the introduced varieties perform poorly because they're often not adapted to the new environment. Sometimes the performance of a variety in the new environment improves with the number of generations grown there. This is known as acclimatization. This process of adaptation is known as acclimatization. And then the multiplication and distribution. Promising introductions or selections from the introductions may be increased and released as varieties after necessary trials. And most of the introductions, however, are categorized for desirable traits and are maintained for future use. And such materials are used in crossing programs and are readily supplied by the Bureau on request, either to the farmers or research workers, scientists, or any research institutes as such. And the next one is, what is the purpose of introduction? What are the various merits, demerits? And if we go into the purpose of introduction, we have various various needs of introduction. They are being outlined like this. And in order to obtain some entirely new crop plant and to obtain new crop varieties, and also these obtained varieties can be used in crop improvement programs, and also in certain areas these crops may be affected by disease and pests, but in a newly introduced area they are protected against these pests and diseases. So in order to save the crop from the pests and diseases is one of the purpose. Then again, for scientific studies, for aesthetic value, and also varieties can be selected from introduction. Selections can be done, pure line, mass, anything. Then varieties are also developed through hybridization from these introductions. These introduced varieties can be used as parents in hybridization programs. And there are also few merits, which can be like they provides entirely new crop species to a particular country or an area. Entirely new crop species can be introduced, and also new they can be released as new varieties, the crop directly as a new varieties. And this introduction and exploration, it's a very feasible method of plant breeding for improvement. It is very quick and also economical, um, and it protects variability from genetic erosion, and then it's very quick and economical, and particularly when the introductions are released as varieties either directly or after a simple selections. Then plants may be introduced in new disease-free areas in order to protect them from damage. Example: coffee, rubber, which were introduced into new areas in order to protect from new various diseases. And uh, like um, coffee was introduced in South America from Africa in order to prevent losses from leaf rust, and similarly rubber was brought to Malaya from South America in order to protect from leaf diseases. And the next one, demerits, obviously we have again introduction of plants, which again introduction of plants may carry new diseases, weeds and pests along with the introduced crop species or variety, etc. Like it acts as a carrier of new weeds, diseases and pests, etc. Thank you.